Hello, welcome to the new session of Biomolecules NCERT based question. This is the second part and we are going to cover the whole chapter in this particular part. You will find NCERT based question in a mixed form in this particular session. So let us start with the question. The first question is, building block of nucleic acid is. So how the nucleic acid is formed? Nucleic acid, if you remember, DNA and RNA. These are the two nucleic acid present. So nucleotides, nucleoside, amino acids, fatty acids. So amino acids are the monomer which are, or we can say the unit which used to form proteins. Fatty acids required in the formation of fats. Now, there is a difference between these two options, nucleotide and nucleoside. If you remember what is the difference, then you are able to give this answer. So, I'm just taking this two option, nitrogenous base. I'm just writing N base, sugar, and phosphate group. Okay, so nitrogenous base and sugar, this form nucleoside, and nitrogenous base and sugar when get attached to the phosphate group means nucleoside when get attached to the phosphate group, it forms nucleotides. This forms nucleotides. So this is the building block of the nucleic acid, nucleotide. So the correct answer is nucleotide. Next question. Sugar phosphate backbone in nucleic acids. How the sugar phosphate backbone is formed? Sugar phosphate backbone in nucleic acid is hydrophilic in nature, it is negatively charged, or show polarity, or all of the above. So it is hydrophilic in nature. This is hydrophilic in nature. Phosphate is an hydrophilic in nature. Sugar phosphate group, it is hydrophilic. It is negatively charged. The phosphate group is having PO4. It is having negative charge. Okay. And the DNA is overall is having a negative charge over it. So this is due to the phosphate group. So it is having negatively charged. It show polarity. So all of the above is the correct answer. Shows sugar phosphate backbone shows this is hydrophilic in nature, it is negatively charged, and it also shows the polarity. Next question. Nucleotide is formed purine, pyrimidine, and phosphate group. Purine, sugar, and phosphate group. So here when you write purine, pyrimidine, both are nitrogenous base group. This is nitrogenous base. There are the two different types of nitrogenous base, purine and pyrimidine. Purine, sugar and phosphate. So only purine, sugar and phosphate they are saying. Next option is nitrogenous base, sugar and phosphate. Pyrimidine, sugar and phosphate. So when you write pyrimidine, sugar and phosphate, so you are discarding purine in that case. And when you write purine, then you are discarding pyrimidine. But both the nitrogenous base are present. In the nucleotide formation, whether it is a purine or pyrimidine. And the two other requirements for the nucleotide is sugar and phosphate group. So the correct answer is C. Nitrogenous base, sugar and phosphate group. These three unite together to form the nucleotide molecule. The two polynucleotide chain in DNA are, this is very important when you draw a chain. Suppose this is one strand or the helix of the DNA. This is 5 prime to 3 prime. So the another one is, this end will be 3 prime and 5 prime. So this is showing the parallel, but this is anti-parallel. One is going to this direction, 5 prime to 3 prime, and if we draw this 5 prime to 3 prime, so this time the direction is in this. So it is parallel, it is discontinuous, it is anti-parallel, it is semi-conservative. The two polynucleotide chain in DNA are, synthesis of this DNA is semi-conservative. But here it is asking about the two polynucleotide chain in the DNA are. So it is anti-parallel in nature. It is not discontinuous, it is continuous. So this is incorrect. It is parallel, it is anti-parallel because they are having the polarity, and here the poles are different at both the ends. Okay, so it is anti-parallel in nature. Next question, which of the following statement is correct? The first statement, adenine pair with thymine through one hydrogen bond. So adenine pair with thymine with one hydrogen bond. Adenine pair with thymine with 3 hydrogen bond. Adenine pair with thymine with 3 hydrogen bond. Adenine does not pair with thymine. So adenine do not pair with thymine. Adenine pair with thymine through 2 hydrogen bond. So if you remember that adenine always pair with thymine. Adenine never pair with, there are 4 different types of nitrogenous bases, namely adenine, thymine, guanine and cytosine. So adenine always pair with thymine. It never pair with guanine or it never pair with cytosine. So this is the third option which is incorrect. Now, adenine, if you remember, adenine always pair with thymine with 2 hydrogen bonds, and guanine always pair with cytosine, or cytosine always pair with guanine with 3 hydrogen bond. So the first one, that is adenine pair with thymine with 1 hydrogen bond, this is incorrect. Adenine pair with thymine with 3 hydrogen bond, this is also incorrect. Adenine pair with thymine with 2 hydrogen bond, this is the correct option. So our answer is correct, the D1, adenine pairs with thymine with 2 hydrogen bond. Next question, which one of the following pair of nitrogenous bases of nucleic acid is wrongly matched with the category mentioned against it? Adenine and thymines are the purines. If you remember, there are 2 groups of nitrogenous bases, purines and pyrimidines. Adenine, thymines are purines. Thymine and uracils are pyrimidine. Uracil and cytosine are pyrimidine. Guanine and adenine are purine. So adenine and guanine are purine, and cytosine and thymine are pyrimidines, and in case of RNA, instead of thymine, uracil is present. So cytosine, thymine and uracil, these are the 3 pyrimidines. So the correct answer is adenine, thymine are purines. Actually, this is a wrong match, and here in the question it is also asked the wrong match. So if you see thymine, uracil and cytosine, these are the pyrimidines. Uracil and cytosine, again they are the pyrimidines. Guanine and adenine, these are the purines, but adenine and thymine, these are not purines. Thymine is not a purine. Okay, So the correct answer is adenine and thymine are purines. Nucleotides are the building block of the nucleic acids. Each nucleotide is composite molecules formed by base, sugar and hydroxyl group, base, sugar and phosphate group, sugar and phosphate group, base, sugar and acid. So base, sugar and hydroxyl, there is no hydroxyl group is present in the nucleotides. Base, sugar and phosphate, this is the correct one, that forms the nucleic acid. Base and sugar unite together to form the nucleoside, but when it joins with the phosphate group, nucleoside when joined with the phosphate group, it forms the nucleotides. Sugar and phosphate group, they don't form the nucleotides. Base, sugar and acid is not present in the nucleotides. So the correct option is D, that is base, sugar and phosphate. Moving to the next question. The quaternary structure of the protein has minimum 1 polypeptide chain, 2 polypeptide chain, 3 polypeptide chain or 4 polypeptide chain. Here they have asked minimum number of polypeptide chain required to form a quaternary structure. So if we form a quaternary structure, then we require, this is one subunit of tertiary structure. And similarly, we will having one more subunit, second subunit. That subunit can be same or different. So when these two subunits unite together with the help of some bond formation like hydrogen bonds, they form the quaternary structure. They form the quaternary structure. So at least 2 subunits they require. 2 polypeptide chain, at least 2 subunit or the 2 polypeptide chain require. One polypeptide chain can only reach up to the tertiary structure. Okay, no. So one polypeptide chain cannot form the quaternary structure. They can reach up to the tertiary structure only. 3 polypeptide chains. So they have asked the minimum number of requirement of the polypeptide chain. So the correct answer is 2 polypeptide chain. They can be formed. 3 polypeptide chain can form a quaternary structure. 4 polypeptide can also form the quaternary structure. But the minimum number required, that is 2 polypeptide chain. So the correct answer is 2 polypeptide chains required to form the quaternary structure. In a polypeptide, the first amino acid is represented as N-terminal on the left hand, N-terminal on the right hand, C-terminal on the left hand and C-terminal on the right hand. So if you remember, we are having a polypeptide chain. So always this is the first amino acid. If this is the first amino acid, then it will having this NH2 group, then it will be having here COH group, then R, then H. So here, this is the left hand, this is the left hand, and this is the right hand of this polypeptide. Right hand of this polypeptide, and here it will be C, at the end it will be having C-terminal. C-terminal. So the correct answer is N-terminal on the left hand. N-terminal on the left hand. Any polypeptide chain, it will have N-terminal on the left hand and C-terminal on the right hand. For a protein to have a quaternary structure, it must what? Have 4 amino acids, consist of 2 or more polypeptide subunit, consist of 4 polypeptide subunit, have at least 4 disulfide bridge. 4 amino acids. Incorrect. Actually, the quaternary structure require subunits, and that subunits must reach up to the tertiary level. That is, it is a polypeptides. So at least they have consist of 2 or more polypeptide subunit to form a quaternary structure. So this, if you write, the consist of 4 polypeptide chain. So there must not be 3, or there must not be 5 polypeptide subunit. So this is incorrect. Okay. It may consist of 4 polypeptide subunit to form a quaternary structure, but it consists of 2 or more polypeptide subunit. This is more appropriate. And have at least 4 disulfide bridges. This is not a criteria to form a quaternary structure. Okay. So polypeptide may have these disulfide bridges, but this is not a criterion to form a quaternary structure. So the correct option is B, that is, it must consist 2 or more polypeptide subunit to form a quaternary structure. Next question. The quaternary structure of a human hemoglobin is best described as a dimer of identical subunit, dimer of a different subunits, tetramer of 4 different subunits, tetramer of 2 different subunits. So if you remember the structure of a human hemoglobin, then it is having 2 alpha and 2 beta. So 1 alpha, 1 beta, and here 1 alpha and 1 beta. This forms the human hemoglobin molecule. So there are 2 different subunits. We are having 2 different subunits, alpha subunit and beta subunit. But what is their number? 2 plus 2, 4. That is the tetramer of 2 different subunits. So the correct answer is tetramer of 2 different subunits. The secondary structure of that portion of an integral protein, where the integral protein is present, if we remember the structure of a plasma membrane. I'm just drawing a structure of a plasma membrane, a bit of it, to understand the intrinsic protein and or integral protein. So this is a bilipid layer. This is a lipid layer, and here a protein molecule is there. Again here. Now here we can see some external protein is there. Okay, so this one is integral protein. Integral protein. So coming to the question, the secondary structure. Now we are talking about this protein secondary structure. This protein secondary structure. The secondary structure of that protein, of an integral protein, that is buried in the lipid bilayer of a cell membrane is. So we are talking about this. We are talking about this integral protein secondary structure. So what type of structure they are having? They are having B strand, or we can say the beta strand, B bend, random coil, or alpha helix. So this secondary structure, this particular secondary structure is having a helical structure, and this is the alpha helix structure. Okay, so the secondary structure of that portion of an integral, that portion, this not the outer portion, the integral protein that is buried in the lipid bilayer, it is having alpha helix structure. It is having alpha helix structure. So this is the correct option. Option D is the correct one. Moving to the next question. Select the incorrect statement. Let us read about the fourth statement. Ribozymes are the nucleic acid with a catalytic power. Proteins are homopolymer made up of amino acids. Inulin is a polymer of fructose. And glycogen is stored in the liver and muscles. So let us discuss one by one the options. Ribozymes, these are the enzymes which are having, actually these are the nucleotide. RNA, when having an catalytic activity or enzyme activity, it is called as ribozyme. So this is the correct statement. Nucleic acid with the catalytic power. RNA is a nucleic acid. RNA is a nucleic acid. So when it is having a catalytic power, it is called as ribozymes. Proteins are the homopolymer made up of amino acid. When you write this, homopolymer means your monomer is same. Your monomer is same. And our polypeptide is formed by amino acids. But we are having 20 different amino acids. So our monomer is not same. Our monomer is not same. These amino acids unite to form polypeptide. So this is not a homopolymer. This is heteropolymer. This is heteropolymer. So this is incorrect. First one is correct. So you got the answer. But let us discuss the C and D option also. Inulin is a polymer of fructose. This is correct one. Inulin is found from the beta root and beetroot, and it is a fructose. It is a polysaccharide made up of fructose. So this is also a correct statement. Glycogen is stored in liver and muscles. So glycogen is a human starch, or you can say animal starch. It is stored in liver and muscle. So this is also the correct option. So protein are the homopolymer made up of amino acid. This is a incorrect statement. So the correct one is proteins are the this is a correct option. The correct statement must be protein are the heteropolymer made up of amino acids. Next question. If all the peptide bonds of a protein are broken down, then what would we remain? Amino acids, peptides, polypeptides, oligopeptides. So you know that amino acids join together with the help of peptide bonds and form a polypeptide. So this is a polypeptide. And when these bonds are broken down, our polypeptide structure is degraded. So this is incorrect. Oligopeptide. Oligopeptide is what? 1 to 10 amino acid unite together to form an oligopeptide. So this is also incorrect. All the peptide bonds are broken down. So 1 to 10 cannot be formed. Peptides. These are not peptide. What is remain here? These are amino acids. So the correct option is amino acids. Next question. Find out the odd one from the given pair. Polysaccharide glycosidic bond means polysaccharide. In polysaccharide formation, glycosidic bond is involved. Protein, for the formation of protein, peptide bond is involved. Fat formation, ester bond is involved. Polynucleotide, hydrogen bond is involved. So coming to the first one, polysaccharide glycosidic bond. If you remember, polysaccharides units are formed by the formation of the glycosidic bond. Monomers are united together or they form a bond, and these bonds are formed as glycosidic bond. If you remember the structure of a starch or a glycogen, then you must remember they are having alpha 1,4 glycosidic linkage or beta 1,3 glycosidic linkage. So there are the different types of bonds at the bond formation, at which carbon is involved in the formation of bond. The name is given according to that. But they are all glycosidic bond. So this is the correct option. Protein formed peptide bond. Yes. Amino acids are the unit that are united in a chain form with the help of peptide bond. These are the peptide bond. Fat, they form ester bond, and that is called as esterification. That reaction is called esterification. So this is also correct. Polynucleotide. Suppose we are having one polynucleotide, that is base, sugar, and phosphate group. Again, we will having here a phosphate group, sugar, and base. So this is one nucleotide and this is the second nucleotide. These nucleotides are joined here by the phosphodiester bond. Phosphodiester bond. And this forms the polynucleotide, not the hydrogen bond. Hydrogen bond is present between the complementary base pair. So if here it is A, then here it will be T, and hydrogen bond will be present in between these two nitrogen bonds. So that forms the double helical structure. But this is one polynucleotide, and this polynucleotide is formed by the formation of the phosphodiester bond between the nucleotides. So this is the odd one out. So the correct option is option D. Okay, moving to the next question. The bond between the nitrogen base and the sugar in the nucleic acid. So if we form a nucleotide, then there is a sugar molecule, and with this a nitrogenous base is attached here. Nitrogenous base is attached. Suppose this, and here this is the sugar. And with this hydroxyl group, phosphate groups are attached. So this bond, this is a glycosidic bond. Glycosidic bond. So in this question, this is asked, the bond between the nitrogen base and sugar in the nucleic acid or the nucleotide is a glycosidic bond. So the first option is the correct glycosidic bond. Anti-parallel strand of a DNA molecule means. So what does this mean? Anti-parallel strands of DNA. So the first option, the phosphate group of the two DNA strands at their end share the same position. The phosphate group at the start of the two DNA strands are in the opposite position. One strand turns clockwise and one strand turns anti-clockwise. So if I draw the helix of a DNA, or we can say the DNA polarity structure. So if this is the 5 prime, this is the 3 prime, and if this is the second strand which are complementary attached, then this is the 5 prime and this is the 3 prime. So this is anti-parallel strand. This is the anti-parallel strand of DNA. So here if we talk about this particular end, this is the 5 prime, then this will be the 3 prime, and this is the 3 prime, then this is the 5 prime. So the phosphate group at the start of the two DNA strand are in opposite direction. This is showing the opposite direction. So the correct option is the option B. The phosphate group at the start of the two DNA strands are in the opposite position. Next question. The pairing of adenine thymine and guanine cytosine GC is the universal phenomena of the DNA. Everywhere you can see in the structure of DNA that A always pair with the thymine and G always pair with the cytosine, or we can say thymine always pair with the adenine and cytosine always pair with the guanine. Okay. Now from this we can deduce the following except, means this particular thing favor all the three option except one. Now we have to find out that one option that do not get fit according to this. A plus T is equal to G plus C. Second option, ratio of A is to T is 1. Guanine equals to cytosine and nitrogenous base in the two strands are complementary. So let us take the D option first. Nitrogenous bases in the two strands are complementary, means A will pair to T and G will pair to C, C will pair to G and T will pair to A. In this way, you will find the complementary. They are the complement to each other, nitrogenous bases. Okay. Guanine equal to cytosine. So guanine is pairing with the cytosine. Always the number of guanine will be equal to cytosine. Guanine never pair with adenine or a thymine. So the percentage of guanine will always be equal to the percentage of cytosine. So guanine equal to cytosine. In the same way, ratio of A is to T is 1. It means that adenine will always pair to thymine. So the percentage of adenine will always equal to the percentage of thymine. Okay. So suppose there are 20% adenine is present in a sample of DNA. So 20% of adenine, then 20% of thymine also. So if you find a ratio, you will get 1. So this is also correct. A plus T is equal to G plus C. So this is according to the Chargaff rule. Chargaff rule says that purine is equal to pyrimidine. Percentage of purine is equals to percentage of pyrimidine. So purine is adenine and guanine and pyrimidine is cytosine and thymine. So this is incorrect. So the correct option is A plus T is equal to G plus C. This is incorrect. Next question. The backbone of the strand DNA is formed by, this is very important question, sugar, nitrate, sugar chain, sugar and nitrate that forms the sugar chain, sodium and phosphate that forms the sodium chain, sugar, phosphate that forms the sugar chain, sodium, potassium, sodium chain. So the backbone, if we see the nucleotide is formed by, this is phosphate group, this is sugar group, and this is a nitrogenous base. So this is one nucleotide. Now again I am drawing one nucleotide. So they are formed by the phosphodiester linkage. They are connected, and this particular, like this particular structure form the backbone of the DNA. So the correct option is sugar phosphate. Sugar phosphate, sugar chain, that is the backbone of the strands of DNA. Next question. Choose the correct statement. There is no uncatalyzed metabolic living system. First option. The living state is an equilibrium steady state. The rate of a chemical process double or decreases by half for every 37 degrees centigrade change in either direction, both B and C. Okay, so which is the correct statement? There is no uncatalyzed metabolic living system. Every metabolic living system is catalyzed by enzymes. So this is the correct. There is no uncatalyzed. So living state is an equilibrium steady state. It is not a steady state. So this is incorrect statement. The rate of a chemical process doubles or decreases by half or every 37 degrees centigrade. This is the normal room temperature. And when you doubles it, the reaction, chemical enzyme catalyzed reaction never doubles or decreases. At that higher temperature, the enzyme get degraded. So there is no question of increasing the rate of reaction over there. So these two statements are incorrect. So the correct statement is there is no uncatalyzed metabolic living system. Next question. Which of the following statement is correct? All biomolecules have a turnover. Turnover means that the molecules are randomly changed into some other biomolecules. Turnover means that the molecules are randomly changed into some other biomolecules. All of the statements are correct. So, let us understand what is the turnover. Turnover is the fixed number of biomolecules that are formed by enzyme catalyzed reaction. So that is fixed, that this particular substrate will form this number of product, and that is fixed for every biomolecule. So it is not a randomly change. Okay. Some or other biomolecules, this particular substrate will form in a catalyzed reaction to form this particular product. That is also fixed. So this is also incorrect. These two statements are incorrect. Okay. So all biomolecules have a turnover number. Which of the given option is correct for the following? The metabolic pathway in which the acetic acid is converted into a cholesterol is an exothermic one. Anabolic pathway generally releases energy while catabolic pathway consumes energy. Without metabolism, there cannot be a living state. Glucose is converted to acetic acid in our skeletal system. So coming to the first option, metabolic pathway in which acetic acid is converted into cholesterol is an exothermic one. Anabolic pathway generally releases energy while catabolic pathway consumes energy. Without metabolism, there cannot be a living state. And glucose is converted into an acetic acid. So these two statements are totally incorrect. Okay, for an anabolic pathway, you require energy. It doesn't releases energy. Catabolic pathway releases energy. Okay, respiration is an example of a catabolic pathway. So in that, energy is released. In that, energy is not consumed. So this is totally incorrect. Okay. Glucose is converted into acetic acid. This is incorrect. Glucose is converted into lactic acid in our skeletal muscles. Without metabolism, there is not be a living state. This is a correct statement. Okay. So we will meet one more session to discuss about some more NCERT based question. Till then, thank you very much.